Hello all, welcome to Life Science Library. In this video we are going to see about another topic from plant physiology, that is phytochrome. It belongs to Unit 6 of Iicsirnet Life Science portion. First, in the past video we have seen about introductory video on photomorphogenesis. In this video we will be seeing about phytochrome in detail. Phytochrome is a photomorphogenic pigment. It has the capacity to absorb red and far red light. It can also absorb blue light, but priority is red and far red. It exhibits photoperiodic function. What are photoperiodic function? Photoperiodic function include induction of flowering, chloroplast development, leaf senescence, leaf abscission, and seed germination. These are photoperiodic function. They have chromoproteins. These chromoproteins occur in dimer. Each component in a dimer have 125 kilodalton. And this chromophore are dimer and they are bound by covalent bond. Chromophore has covalent attachment. This is the introduction about what is chromophore, what are the nature of chromophore, and what are the components of chromophore. Next we are going to see about phytochromobilin. This is the chromophore attached with phytochrome. It is a linear tetrapyrrole ring. It has two components, that is apoprotein. This chromophore, entire phytochrome, has two components, that is apoprotein and holoprotein. Apoprotein is the polypeptide chain. Holoprotein, this phytochrome chain, will attach to chromophore. So, these two polypeptide chain are apoprotein. When the chromophore comes to attach with phytochrome, it is holoprotein. Now we will see the components of this chromophore structure. Dealing with the components of chromophore structure, we have, as usual, we have N-terminal and C-terminal. N-terminal end, we have PAS domain. Next to PAS domain, we have GAF domain. Then PHY domain. Then in the C region, we have HKRD domain. It is connected with PRD and PRD, two PRD domain. These two things are connected with one another by hinge connection. This is the basic chain of phytochrome, polypeptide chain, single polypeptide chain of phytochrome. We have PAS domain, GAF, PHY, two PRD domain and one HKRD domain. This is N-terminal side and C-terminal side. N-terminal side have PHY, C-terminal side have PRD and HKRD domain. This is the basic structure. Now we will draw about the dimer. N-term, we do it occur in dimer. So N-terminal, we will draw two chains. N-terminal, we have PAS domain. Next PAF, we have GAF domain. Next to GAF, we have PHY domain. C-terminal end, we have HKRD domain. It is connected with PRD and PRD. These two regions are connected with hinge region. We have dimer, two things. So let's repeat fastly. PAS, GAF, PHY, hinge region, PRD, PRD and HKRD at C terminal. These two chains are linked with, these two chains have a linkage of covalent molecule, covalent bond. The dimer, it is attached with covalent bond. Now in GIF domain is attached with, the both GIF domain is attached with phytochromobulin. Is attached with phytochromobulin, and here also GIF domain is attached with phytochromobulin. This linkage is thioether linkage. Thioether linkage. This is a previous year question. N-terminal side is attached with phytochromobulin. This is a previous year question. Covalent bond. This is important in CSIR point of view. This occur in dimer. This is a previous year question. Next we are going to see about the nature of these phytochromes. The nature of the phytochrome. This chromophore is autocatalytic and it occurs spontaneously. Phytochrome can be found in two photoreversible form. One is red light absorbing at 666 nanometer in PR form. Far red light 750 nanometer in PFR form. This is a previous year question. In 666 nanometer PR form, 730 PFR form. This is a previous year question. Now how the photo conversion takes place, we will see. PR form and PFR form. These are reversible events. PR form is converted into PFR form in presence of red light. PFR form is converted into PR form in far red. This conversion also takes place in during dark time. It is called as dark reversion. This is also a previous year question in CSIR exam. Please make a note of it. PR is converted into PFR by red light. PFR is converted into PR by far red light. This is a previous year question. Now we are going to see what will happen once the phytochrome, consider this is a phytochrome receive light. What will happen if phytochrome receive a light? This phytochrome will undergo conformational change. It will undergo a conformational change. PR chromophore undergoes cis-trans isomerization. Cis-trans isomerization in C15 end and C17 double bond. This is the basic phenomenon for any reaction that receives an external stimulus that undergoes a conformational change. This is a basic phenomenon. It is a reversible process. Now, we are going to see about the amino acid attachment. Consider this as a polypeptide and this as another polypeptide. How does this trans configuration takes place? Serine, cysteine, histidine. This entire component is attached with cysteine. This is a important pointer in CSIR exam. Cysteine, CYS amino acid in the polypeptide attached with this phytochromobilin. Cysteine. This important previous year question in any competitive exam. This attachment is serine, cysteine, histidine. Linked with phytochromobilin. This attachment is covalent attachment. It is a cysteine is an amino acid. And this cysteine amino acid is this thing takes place in GAF region at N terminal of phytochrome. The proportion of phytochrome in PFR form after saturating the radiation by red light is only 85%. So it is said that these two spectrum will overlap. Very small amount of far red light is absorbed by PR, make it impossible to convert PFR to PR. So equilibrium for PR is 97 and PFR is 3%. This condition is called as photostation. This case is called as photostationary state. There are 5 types of phytochromes: A, B, C, D, E. Characteristics of each type based on the characters of light stability and reaction to the light that classified into 2 different types, type A and type B. Type 1. Type 1, phytochrome A belongs to type 1. They are light liable. Their level drops in exposure to light. PFR decrease. Very low fluence response. Very low amount of light. High irradiation is observed. They are continuous with far red light. They degrade with light. Red light. So they are called as light liable, photo liable. Only example is phytochrome A. Next, phytochrome B. They are light stable. They are inhibited by far red light. A, B, C, D, E are the examples. Phytochrome A, what are the function it will do? Help in seed germination under broad spectrum of light. Seedling de-etiolation. Promote flowering. They don't have any nuclear localization signal and they are light induced. Meaning, in the mode of mechanism we will see that SCOP1 will enter into nucleus and they will exit the nucleus based on the presence or absence of light. This doesn't need nuclear localization signal. Phytochrome B. Seed germination continuously in red light. Seedling de-etiolation. Shade avoidance. C. Primary leaf expansion. D and E. Shade avoidance response. Now we are going to see about the mode of action of phytochromes in case of dark and in case of presence of light. First we will see what will happen in the presence of dark. Before saying that, we need to understand few abbreviations. Few components involved in the signaling pathway. First is COP, another is HY5. COP is called as constitutive photomorphogenic 1. It is E3 ubiquitin ligase which are involved in targeting protein for 26S proteasomal mediated degradation. This is the exact definition of COP1. It is nothing but an ubiquitin ligase. It inhibits photomorphogenesis in dark by degrading phytochrome inducing factor. Phytochrome inducing factor is nothing but a basic helix loop helix motif, and it occurs at the C-terminal end. In dark, COP1 is present in nucleus, but in light it is found only in cytoplasm. HY5 is a transcription factor that regulates photomorphogenesis. If the light is present, COP1 will move out into the cytoplasm and HY5 will undergo transcription and translation, and it undergo photomorphogenesis. This is the basic components of signaling in phytochrome. Now we will see what will happen in dark. Dark condition. In case of dark, condition, we have nucleus. Inside nucleus we have COP1 attached with HY5. It is the transcription factor. This undergo, because it is dark, no light, it undergo E3 ubiquitin ligase mediated proteasomal degradation. So HY5, no transcription. No transcription, no photomorphogenesis. No photomorphogenesis occurs. This is the mechanism during dark. During light, if light is present, what will happen in the presence of light? Consider this as a nucleus. We have COP1, HY5. When light is irradiated, COP1 moves outside to cytosol. So this HY5 will remove from COP. HY5 will undergo transcription and give rise to photomorphogenesis. This is the basic idea. Next we are going to see about few related terminologies of photochrome of phytochrome response. One is fluence. What is fluence? Fluence is nothing but number of photons imprinting. It is called as fluence. Next, what is very low fluency response? Because the amount of light needed is very low. Amount of light needed is little high when compared to VLFR, that is low fluence response. This reversible conversion of PR, PFR and this reversible process is coined by Hendrick and Bothwell. They follow a law of reciprocity. What is the law of reciprocity? If the radiation time is low, then the radiation concentration should be high. If radiation time high, response can be given even in dim light. This is fluency. Because this phytochrome is a photon induced signaling, we are duty bound to understand fluency and the law of reciprocity. If high time, response given even in dim light. At low time exposure, the radiation of light should be at very high concentration to get a desirable response from phytochrome. This is the overall idea of phytochrome. Now, let's sum up everything with regard to phytochrome, what all we have seen, and the important thing for this CSIR exam. Phytochrome absorb red and far red. That doesn't mean it doesn't absorb blue light. It also has the capacity to absorb blue light. It is a dimer. Each have 125 kilodalton. It is attached with chromophore. These are important. Next, we have tetrapyrrole ring in the phytochromobilin. Now let's draw what will happen in the N terminal and what will happen in the C terminal. In the N terminal, we have PAS domain, GAF domain, we have PAS domain, we have GAF domain, we also have PHY domain. This is hinge region, covalent linkage. It is attached with PRD domain. It is attached with PRD domain and it is again attached with HKRD domain. It is the C terminal side. This replica will also be seen because it is a dimer and each dimer has, it is a GAF domain and each dimer has 125 kilo, it is a hinge region, 125 kilodaltons, PRD domain, HKRD domain at C terminal. These two dimer are linked by covalent chain. This GAF domain is attached to phytochromobilin by using a thioether linkage. It is attached with a thioether linkage. This is the previous year question. Covalent bond previous year question. Attached to the GAF domain. It is in the N terminal. At the GAF domain, this phytochromobilin is attached with cysteine amino acid. It will undergo conformational change and cis trans isomerization when they are exposed to this kind of red light. They will undergo conformational changes and cis-trans isomerization. It is attached to the GIF domain with cysteine amino acid. These are the basic details. Now we have phytochromes at red light absorbing and far red light absorbing. Red light absorbing 616, far red light 730. This is the previous year question. Then we have 5 types. Type 1 and type 2. Type 1 is light liable, that is phytochrome A. Type 2 is light stable. All other things they are inhibited by far red light. They don't have nuclear localization signal. Then mode of action of phytochrome. COP1, it is a E3 ubiquitin ligase. HY5, it is a transcription factor. This COP1 inhibits photomorphogenesis. So now we will see as every signaling pathway we have seen so far. In case of dark, we will have nucleus. We have COP1 and HY5. As there is no light, these are subjected to E3 ubiquitin ligase mediated proteasomal degradation. So, no photomorphogenesis. When there is a light is present, basically it is a red light. The light is present. Consider it as a nucleus. COP1, HY5. This COP1 is going to cytosol when it is light induced, and HY5 will undergo transcription. We have photomorphogenesis. This is the whole idea of signaling. For this movement, we don't need nuclear localization signal if it is in case of PHYA. This is the whole idea of this phytochrome. Hope this video is very clear, and hope this video is useful for all CSIR aspirants watching this video. Thank you for watching this video. Thank you.